The modern regenerative medicine market presents a strict paradox. Despite an explosion of commercial marketing for cellular releases, there are exactly 0 FDA-approved exosome products available for purchase. The clinical consequences of this disconnect are severe. In December 2019, patients in Nebraska were hospitalized with systemic sepsis after receiving contaminated biological preparations from EUCY laboratories. The subsequent FDA investigation into the manufacturer documented 152 distinct sterility failures, including contamination by 9 separate bacterial species. Regulatory enforcement accelerated in response. Between 2020 and 2025, the agency issued an unprecedented slew of warning letters to companies like Chimera Labs and Evolutionary Biologics for marketing unapproved biologics. The core issue driving this crisis is an absence of data. Products are sold to clinics for thousands of dollars per session, yet the manufacturers have failed to publish analytical characterization proving what the vials actually contain. The global scientific community established a clear boundary. MySev 2023, a consensus guideline authored by over 1,000 contributing scientists, explicitly states, there is a lack of MySev support for uncharacterized cell releasates being described as exosome therapies. In a landscape defined by aggressive marketing and escalating regulatory enforcement, physicians face a clear mandate: demand independent analytical characterization data before treating a patient. AutologIX by Swiss Concept is a point-of-care medical device designed to produce a complete autologous biological concentrate from a patient's own blood in approximately 20 minutes. The system operates on precise physical parameters. It utilizes a 15 kilodalton ultrafiltration membrane, with pores measuring 5 nanometers or less. Because the smallest known exosome is roughly 30 nanometers, retention is a physical certainty governed by the membrane. This diagram represents the 3 core pillars established by the MySev 2023 International Consensus to prove extracellular vesicle identity: quantification, protein composition, and morphology. To address the first pillar, quantification, autologIX employs nanoparticle tracking analysis. The result is a measured concentration of 30 trillion total particles per milliliter. That concentration is roughly 50 to 100 times higher than published levels for standard PRP-derived vesicles. This exponential increase is achieved by concentrating 20 milliliters of plasma down to a 6 milliliter output through ultrafiltration. For the second pillar, protein composition, flow cytometry confirmed high positivity across the 3 canonical tetraspanin surface markers: CD9 at 93.2%, CD63 at 90.1%, and CD81 at 69.1%. For the third pillar, morphology, transmission electron microscopy provides direct visual confirmation of membrane-bound vesicular structures that match established EV profiles in the scientific literature. The minimum MySev requirement is 2 complementary techniques. By executing 3 distinct analytical methods to address all 3 core pillars, autologIX delivers a measured data package that exceeds the basic international standard. This intersecting diagram illustrates why evaluating complex biological fluids requires orthogonal testing. Every single analytical method has an inherent blind spot. Nanoparticle tracking analysis perfectly answers how many particles exist. However, it counts all nanoparticles indiscriminately, meaning it cannot distinguish between true extracellular vesicles and co-concentrated lipoproteins or protein aggregates. Flow cytometry compensates for this by answering what they are. It uses fluorescent antibodies to isolate and tag only the specific population carrying true EV molecular markers. Yet flow cytometry alone is incomplete. While it proves molecular identity, it cannot provide the absolute particle concentrations required for standardizing clinical applications. Transmission electron microscopy acts as a nanometer resolution magnifying glass to answer, do they look right? It visually verifies the presence of a lipid bilayer, though it lacks the ability to quantify its scale. NTA quantifies, flow cytometry identifies, TEM visualizes. Together, these methods provide convergent proof from independent physical principles, cleanly separating scientifically characterized preparations from marketing claims. Shifting from physical characterization to biological function, platelet-derived extracellular vesicles represent 70 to 90% of all circulating EVs in human blood. These vesicles carry an extensive payload, including over 250 identified microRNAs, dual-axis growth factors, and bioactive lipids. This cellular signaling flowchart outlines published therapeutic mechanisms. A 2017 study by Guo and colleagues established that PRP-derived exosomes interact directly with fibroblasts to activate the YAP signaling pathway. Activating this pathway promotes cellular proliferation, migration, and re-epithelialization for wound healing at a rate superior to standard plasma alone. In the context of cartilage repair, 2024 research by Liu and colleagues demonstrated these vesicles activate the PI3K-AKT pathway in chondrocytes, driving the upregulation of critical structural proteins like COL2 and SOX9. For hair restoration, published 2025 data revealed that platelet-derived EVs upregulate beta-catenin in dermal papilla cells, specifically enhancing hair shaft elongation. These regenerative effects are documented in peer-reviewed literature through specific biological pathways, establishing exactly how autologous vesicles operate in vivo. The autologIX system is not an off-the-shelf biologic. It is a point-of-care medical device that processes the patient's own blood directly at the bedside. Utilizing the patient's own blood eliminates the severe clinical risks of allogeneic disease transmission, cross-contamination, and the variables introduced by pooled donor sources. This split screen highlights the immunological advantage. Because autologous EVs carry self-MHC molecules, they integrate smoothly with host cells, bypassing the T-cell activation and immune rejection risks associated with allogeneic cross-dressing. The regulatory pathways diverge sharply. Manufactured allogeneic exosome products require a biologics license application under Section 351 of the Public Health Services Act, the FDA's highest regulatory bar. By contrast, the autologIX device operates under the established 510(k) medical device pathway and the same surgical procedure exception, supported by decades of regulatory precedent for platelet-rich plasma devices. The clinical safety record of autologous blood concentrates is extensive. A meta-analysis of 36 studies covering over 2,000 patients reported 0 serious adverse events related to the injection. The autologous device pathway presents a documented, practical risk profile that avoids the strict regulatory and safety pitfalls of manufactured allogenic biologics. This chart details the 3-tier evidence classification framework. Scientific rigor demands absolute transparency regarding the exact boundaries of the available data. While the analytical data confirms the physical presence and molecular identity of exosomes within the concentrate, this characterization does not constitute clinical proof of therapeutic efficacy for the device output itself. Establishing definitive therapeutic outcomes requires dedicated clinical trials, which are currently in development. The testing parameters must also be stated clearly. The analytical characterization was commissioned by the manufacturer rather than independently peer-reviewed. The assays themselves have hard boundaries. The 30 trillion particle count includes co-concentrated proteins, and evaluating the remaining IMICEV Category 2 and 3 markers requires further testing. These limitations represent strict adherence to the facts. In a market where the majority of products fail to prove their exosomes even exist, transparent, well-documented characterization is where scientific credibility begins. The modern regenerative medicine marketplace is saturated with aggressive marketing language, yet it remains severely starved for objective analytical proof. autologIX addressed this deficit by subjecting its concentrate to the strict physical laws of ultrafiltration. This composite displays the final readout: 30 trillion particles per milliliter, high positivity for CD9, CD63, and CD81, and documented vesicular morphology. 3 distinct analytical methods satisfying the core pillars of one international consensus standard. For the physician evaluating regenerative technologies, the standard has evolved. It is no longer enough to ask if a product mentions exosomes in its promotional materials. You must ask if the manufacturer actually measured them. Clinical confidence requires evidence, not dialogue. In science and medicine, measured always beats claimed.